Assalamu alaikum dear students. Today we are going to discuss what is meiosis. Many organisms pass their gene to their offspring through sexual reproduction. This begins when two gametes unite to form an embryo that is generally unique from the parent organism. The embryo then grows into adult, which turn pass their genetic information into their own offspring. Gametes are formed through a series of processes called meiosis. The cell that undergoes meiosis to produce the gametes are called the germline cell. In diploid organisms, the germline cell have two copies of each chromosome. The germline cell undergoes meiosis to produce haploid gamete, which only have one copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into adult. Meiosis is just one step in the life cycle of the germline cell. Similar to mitosis, the cell also passes through the interphase G1, S and G2 stages before they enter the meiosis. The DNA inside a germline cell is duplicated before meiosis begins. During the S phase, the duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are two cell division events during meiosis. The first division is the Meiosis I, result in two unique daughter cells that have half the amount of the DNA as the parent germline cell. The second division, that is the Meiosis II, result in four unique haploid cells that only have one copy of each chromosome. These haploid cells are the gametes that could go to produce an offspring through sexual reproduction. Let's take a look more closely to each of the division events. Meiosis begins with the prophase I. In this stage, the DNA condenses to form chromosome. Here, we see the duplicated sister chromatin joined together at the centromere. They stay fused at the centromere throughout the Meiosis I. Next, each pair of the homologous chromosome undergoes synapses to form a complex involving two pairs of sister chromatins. The chromosomal material is exchanged between the two pairs of sister chromatin. This event is called recombination, or more commonly crossing over. After the crossing over, the sister chromatin from each chromosome are no longer identical to one another. This is one of the reasons why no two sisters aside from the twin are genetically identical. There are several more keys in prophase I. The nuclear membrane begins to break down. Then the two centrosomes migrate to opposite end of the cell and the microtubule appears. The microtubule then attached to the chromosome. The next phase of the meiosis I is called the metaphase I. Here, the synapse chromosome align at the equator of the cell. The chromosome align randomly, which result in different combination with time meiosis occur. Each time meiosis occur. The next phase is anaphase I. During this phase, the homologous chromosomes separate and migrate to two poles of the cell. Importantly, the sister chromatin remain attached at the centromere. The final step of meiosis I are telophase I and cytokinesis. Here the cell divides into two daughter cells. Each of these two cells now undergoes meiosis II. The meiosis II is similar to mitosis. The first stage of meiosis II is prophase II. Again the chromosome condenses, the nuclear envelope breaks down, and the spindle apparatus form. The major difference between the prophase II and the prophase I is the fact that the daughter cell have only one copy of each homologous chromosome. So in the prophase II, there is no synapses of the homologous chromosome or crossing over. In metaphase II, the chromosome align at the equator of the cell. Again, the alignment is random. Since the sister chromatin are no longer identical, there will be many different possible ways for these chromosomes to align. In anaphase II, the sister chromatin are pulled apart as the microtubules shorten. Also, the end of cell are pushed further apart as the microtubules elongate. In the telophase II, the nuclear membrane reforms and the cytoplasm divide into two haploid daughter cells. This division is called cytokinesis. Since the meiosis II began with 2 cells and each of these cells were split into 2 cells, we now have 4 unique haploid cells at the end of the meiosis. These cells are called gametes. 2 gametes, one from the father and one from the mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis. I hope you really liked the video. Thank you very much for watching.